Thanks.
Thank you very much, Ryan, for agreeing to give a Build a Cell seminar, and you can get started.
Thank you, Kate. Hi, everyone. My name is Ryan. I'm very excited to be here today and give this maybe slightly unconventional Build a Cell seminar here. I figured I'd start by giving a little bit of background on myself, actually. I am an immunologist and a biochemist by training. I did my PhD in a structural immunology lab studying T-cell receptor recognition of self-peptides using protein engineering and X-ray crystallography to investigate how T-cell receptors distinguish self from non-self proteins. Then I worked in vaccine development for a few years before eventually joining the effort to help write the report on the feasibility and risks of mirror last year, which is, of course, what I'm going to be talking to you about today. I'm based in the San Francisco Bay Area, but I'm currently calling in from the UK because I was just at the iGEM Jamboree in Paris last week. I just kind of always like to know where people are. So I also wanted to give a quick intro or like a quick sentence on the Mirror Biology Dialogues Fund. The science article that I'm about to discuss called for more discussion on this topic of mirror bacteria. And so to support that discussion, many of the scientists involved worked to raise some money from a number of science philanthropies to help establish what would become the MBDF, or the MBDF, not the. We're a small organization that's working to support the ongoing conversation and the effort to learn more about these risks and find a path forward to addressing them. So I'm sure many of you have heard a lot about this, so I'll aim to give only a brief overview of the background and the potential risks of mirror bacteria before talking about some recent progress since the report was published last December, and then eventually diving much more into the synthetic biology and how our understanding of the feasibility and potential trade-offs have improved over the last year. I think that's most interesting to this audience. And I'm also actually interested for your input, so hopefully lots of time at the end for questions and comments. Yeah, so for a very quick reminder of some of the basics, many molecules are chiral. They come in mirror image pairs, just like left and right hands. This includes most notably proteins and DNA, which I'll be talking a lot about today. Left and right-handed versions of molecules have identical physical properties like thermostability, but they have very different biological properties because they will interact differently with other chiral molecules, like trying to put a right-handed glove on a left hand. It doesn't really work the same way. So, and biological interactions are highly evolved and highly specific. And so a simple inversion in chirality can often make a huge difference biologically. One of the most famous examples probably is thalidomide, shown here, where one molecule on the left is a safe treatment for morning sickness, and the mirror image version on the right causes severe birth defects. So all life on Earth uses biological molecules of the same handedness for all basic functions. And mirror proteins and mirror DNA are not found in nature, though mirror amino acids are in very rare and limited circumstances, like in bacterial cell walls to some degree, a few amino acids here and there. But this concept where all living organisms use only one of two possible handed forms of chiral molecules is called homochirality. And this concept of homochirality was actually discovered by Louis Pasteur in the 1800s. So way back then, Pasteur posited the possibility of life forms with reverse-handedness, mirrored organisms with the complete opposite chirality of known life. Um, and the simplest viable mirror life form would probably be a mirror bacterium. In isolation, a mirror bacterium would be expected to function identically to its natural chirality counterpart. But mirror amino, for example, mirror amino acid sequences would fold into mirror proteins that were the mirror images of natural chirality proteins of their, the same sequence. But as we'll discuss, they can also be, as a whole, as a cell, the mirror bacteria could have very different properties in the natural world. So the risks of mirror life were discussed as early as 1992 in Science magazine after the first mirror protein was synthesized and showed reciprocal substrate specificity, as expected. Brewster and Laskowski here published a letter that noted the potential risks of mirror life forms and the importance of thinking through the implications before creating them. And others have since raised the possibility and discussed these risks periodically, including a 2010 piece in Wired magazine and a 2012 book by George Church and David Regis, I think it was. Anyway, and the NSF actually later funded 2 grants to Kate here, sorry, call you out Kate, and a few others with the express purpose of advancing towards the creation of mirror cells. And this grant did include, credit to Kate, the other authors in NSF, this grant did include an ethics section with some discussion on the risks, and all of the researchers on that group were a part of this working group. So through 2024, this working group of 38 scientists and biosecurity scholars from about 10 different countries came together to try to investigate the feasibility and risks of mirror life and what to do about them. Ultimately, the group wrote an article on the topic, which was published in Science last December. This group was chaired by Jack Shostak and John Glass. and included synthetic biologists like George Church, Craig Venter, Drew Endy, um, uh, Kate here, a number of other people, and a number of immunologists, including Ruslan Metsitov and Mark Davis, people I know well, and microbial ecologists like Rich Linsky, and various other relevant disciplines, including plant biology, biosecurity, chemistry, etc. I think it's fair to say that virtually everyone in this group was initially pretty skeptical about the feasibility and or the plausibility of the risks from mirror life, but after investigating the available evidence in some detail and for quite some time, became quite concerned. And I think it's important to note that the group includes several researchers who previously held the creation of mirror life as a long-term goal, Kate already mentioned, but have since abandoned it because of these risks. So before talking further about the risks and feasibility, I want to first discuss what some of the possible motivations are for creating mirror bacteria. Um, to date, I think it's probably fair to say that the primary motivation has been pure scientific interest, just be an incredible achievement to accomplish. Being able to reassemble life in mirror image form, which would demonstrate really precise understanding of the essential components of life and demonstrate the ability to create it and manipulate it. However, there are also significant potential benefits to mirror biomolecules, which I'll discuss in a slide or two. Um, some mirror biomolecules have shown potential to bind to specific proteins without triggering an immune response or being broken down by proteases, which increases their half-life. So they can be pretty useful therapeutic molecules. And these are currently made via chemical synthesis, but producing larger molecules can be difficult and costly. So the primary proposed application of mirror bacteria is to use them as bioreactors to produce therapeutic mirror molecules at a lower cost and at a greater scale. For example, mirror aptamer therapeutics, shown on the right here, are short nucleic acid molecules that can bind or inhibit a specific target with high affinity. And these mirror aptamers, also called spiegelmers, invented by Germans, have longer half-lives and are less immunogenic. There are a few other proposed benefits. I don't want to say that this is the only proposed application, but the other proposed ones are, in my opinion, a little more out there, and all of which I think are a little unclear as to how truly useful they would be compared to the biomanufacturing question. So some propose that they might be useful to investigate the origins of homochirality on Earth, but I don't think it's totally clear how much it would reveal about the past, although it certainly would provide some interesting possible experiments. Some have proposed that they'd be useful for carbon sequestration because nothing might be able to consume the biomolecules that mirror bacteria generate, or as a cell therapy chassis because the immune system won't be able to recognize them. But again, in my opinion, I think it's worth highlighting that either of these only really work if the worst end of the risk scenario plays out and mirror bacteria can't be consumed in the environment or recognized by immune system. And so if these are possible useful applications, the risks might be worse and change that risk benefit calculus. So in my opinion, I think the most likely useful proposed application is to manufacture mirror biomolecules. So here I just want to highlight one example of a beneficial mirror peptide therapeutic as an example. So you can see here on the right, this is a mirror image mini protein in purple. It was designed, purple here, to inhibit the VEGF protein in humans as a potential cancer treatment. And this was developed via mirror phage display. So a D version of VEGF-A here in green was synthesized chemically as a target for natural chirality peptide inhibitor phage display. And once an effective natural chirality peptide inhibitor in yellow was found, this could in turn be just synthesized in mirror form in purple here, and it will automatically, by principle of symmetry, bind to the natural chirality VEGF protein circulating in the human body. And so, as I said, the two main advantages for developing mirror therapeutics are these increased biostability and decreased immunogenicity. So the mirror peptide is shown here with data plotted in white compared to the natural chirality version in black. And you can see that it has a much longer half-life in the body due to protease resistance. And it does not result in anti-drug antibodies compared to the natural chirality version. So there are already several mirror nucleic acid drugs in early clinical testing, for instance, and there are two FDA-approved peptide drugs that contain all or almost all mirror amino acids. There are other applications of mirror molecules as well, but just highlighting a key one here, often discussed. So I quickly want to define mirror bacteria and how to get there a little bit more before talking about the risks. The creation of mirror bacteria is not imminent, perhaps, but it is becoming increasingly feasible as a variety of technologies advance. So based on our analysis, the most likely pathway would involve combining work on building synthetic cells on one hand, and work on efficiently constructing mirror image versions of existing biomolecules on the other hand, so like DNA and proteins. So these two areas of research could be combined to create a mirror bacterium in one of two ways. The first is bottom-up assembly of mirror biomolecules into a new mirror bacterium. So this is literally assembling the pieces of a cell from non-living parts and then booting them up into a living mirror organism. And the second potential method is a top-down or stepwise conversion of a natural chirality bacterium into a mirror bacterium. This would probably be extremely difficult. It would essentially require building a completely orthogonal mirror biochemistry system uh, inside of a normal cell and then somehow switching it over from the natural chirality system to the mirror system. And from most conversations that we've had so far, it seems like the bottom-up pathway is expected to be more feasible than the top-down in the near future. So I have to express or emphasize that the bottom-up pathway is still extremely difficult. Um, but this talk is going to focus mostly on the bottom-up, although I'll talk about both a little bit later on. Importantly, no known researchers are actively working to make mirror bacteria, but some progress toward mirror bacterium has been made already. I don't need to tell this group that a lot of progress has been made, but there's still a lot left to do. Synthetic biologists have synthesized full bacterial genomes. Recent advances in chemical synthesis are at the point of creating mirror versions of key proteins, including DNA and RNA polymerases, as well as kilobase length mirror DNA and RNA. But many technical barriers remain. For instance, scientists haven't created yet a functional mirror ribosome or a crossover ribosome or related machinery that could translate natural chirality mRNA into mirror proteins. Um, and because of these hurdles and others, it's hard to predict when mirror bacteria might be created. It's always hard to tell the future. But one way to think about this possibility is through costs. So one approximate number that Drew Endy put forward, I hope he doesn't mind me sharing, is it's possible that by default, it might be plausible to create mirror bacteria in roughly 10 to 30 years time without focused efforts, but it could be sooner with like a deliberate investment, somewhere in the range of 500 million to $1 billion. So quite a lot. And this number just sounded reasonable to others in the working group, I think, but nope. Where'd it go? Here we are. And then again, with the theme of the future being difficult to predict, AI systems are also difficult to predict, and their effects are difficult to predict, but they could potentially speed up the development of precursor technologies in unforeseen ways. It could further enable or accelerate the development of mirror bacteria, probably through optimizing certain technical steps, such as protein design or automating things that require automating. So quickly, I just want to go through the risks pretty briefly. So assuming a mirror bacterium is made, I'd like to start talking about the human immunology risks potentially. So first question is how would a mirror bacteria even get inside humans or other multicellular vertebrate organisms? Normally, bacteria use specialized virulence factors to help them invade and enter host tissues and access bloodstream and nutrients. Mirror bacteria would not have functioning versions of these virulence factors. They're protein-based and would not be expected to work even if they had them. But it turns out actually that bacteria already frequently translocate across barrier tissues through mucous membranes or microabrasions, and data shows that virulence factors aren't essential for bacteria to access host organisms. I'm not showing that data here. I'm just going to try to keep the data like really minimal here and go through this somewhat quickly. I apologize, but once inside, evidence suggests that mirror bacteria could evade many key aspects of the immune system. So in humans, I'm just going to show two slides on human immunity. In humans, the immune system can be split into an innate and an adaptive immune system. So I'll speak briefly to each in turn, showing really only a minimal amount of data again. So the innate immune system relies heavily on these specialized pattern recognition receptors that bind to particular pathogen molecules. For bacteria, classical examples are flagellin, LPS, CpG DNA, lipopeptides, peptidoglycan, all of which are chiral molecules. So theory and evidence suggests that these would fail against mirror image. These receptors would fail to recognize mirror image versions of these biomolecules. However, these are pretty easy experiments to do, and not all of these have been tested. So I think more research can and should be done into mirror versions of these molecules. But studies of mirror biomolecules so far have found they do not activate the expected receptors. And I have a question. Did people ever test immune responses in vivo, or was it all in cell cultures? Definitely have been quite a few in vivo uh, tests. The data I'm showing here is not a test of a mirror molecule, but it is a test. I'll explain in a second. Point is, yeah, some mirror molecules have been injected, quite a few mirror proteins that are non-immunogenic, and mirror nucleic acids as well, and they're non-immunogenic. So uh, to go back to the innate immune system, these faulty innate receptors are extremely important, and not having the impaired function is extremely dangerous for an organism. So mice that have a genetic defect in only one of these, TLR2, which binds lipopeptides, bacterial lipopeptides, um, so they still have a whole host of other innate immune receptors and an adaptive immune system. But if they're missing one of these immune receptors, TLR2, they're highly susceptible to bacterial infections, and have much higher mortality rates. Mirror bacteria could plausibly evade TLR2 signaling and many other innate immune receptors signaling pathways as well, um, which I think is potentially concerning. Or quite concerning. So in contrast to the innate immune system, the adaptive immune system is theoretically capable of binding to a very wide range of pathogens, almost anything. Antibodies can be raised against almost anything, including mirror bacteria or mirror proteins that could be found on their surface or other molecules. But unfortunately, its activation and antibody development depends on upstream processes that are chiral, and it's possible that these won't be effectively engaged in a mirror bacterial infection. Um, for example, as I've mentioned, studies show that mirror proteins and peptides are not cleaved by natural chirality enzymes and are non-immunogenic. So this resistance to proteolytic cleavage could heavily disrupt the ability for the immune system to process these antigens and present them to the adaptive immune system, which is what the adaptive immune system uses to recruit T cells, activate B cells, and fight infections. So this is a mirror protein on the bottom here. There's no measurable cleavage compared to a natural chirality peptide. And without T cell activation, antibodies can still be raised, but they're much, much weaker. So research shows here that while natural chirality proteins readily elicit an antibody response from the adaptive immune system, mirror image versions of the same proteins elicit virtually no response over time. And in fact, they're difficult to induce a response to even when trying very deliberately. So there's a rough but informative, I think, clinical analogy here, which is patients with impaired MHC2 deficiencies. These patients are extremely susceptible to infections and most die in childhood, even with bone marrow transplantations. MHC2 is a class of molecules that presents antigens to T cells to stimulate antibody responses. So if mirror protein cleavage is impaired, one would expect that the peptides can't be presented on the MHC2 molecule. And you could expect a similar lack of function of the adaptive immune system. And so again, these are patients that have the whole host of innate immune receptors too. So while there are plenty of caveats to this experiment, it's not an experiment. This is unfortunately just observational data in human clinical trials or clinical situations. Mirror bacteria could plausibly be worse than this if they're evading an adaptive immune response and also the innate immune response. But again, this remains unclear, and there's quite a bit of uncertainty here. Two other analogies that I like to some extent are biofilms and some cancers, both of which are classes of cell types that sacrifice growth potential for immune shielding, which may or may not be a similar analog to mirror bacterial infections, which would presumably have less access to nutrients, but also and pretty extreme immune evasion potential. Finally, just on a quickly note, without showing any data, that while we have a much better understanding of the immunology of vertebrate species than we do other species, it appears that many invertebrate species and potentially plants also, unsurprisingly, have more basic immune systems that could be at least as or more vulnerable to mirror bacterial infections. So while a lot of uncertainty remains here, I think mirror bacteria could be dangerous to an unusually wide range of species compared to other pathogens. Now just one slide on some environmental risks before proceeding. Normally, bacterial populations in the environment are kept in check in large part by bacteriophages and by bacterial predators like protists. However, viruses and bacterial predators rely on chirality-specific mechanisms to function and predate bacteria. Mirror bacteria would certainly be resistant to infection by phages due to the incompatibility with their central dogma on a fundamental level. And it's also likely that they could be resistant to other predators by evading consumption and likely digestion and metabolism too, which could give mirror bacteria a pretty major environmental or ecological advantage. So on the other hand, mirror would probably lack the ability to consume most natural chirality nutrients like glucose, for example. So they'd therefore be dependent on achiral nutrients, which are present in the environment, but to a limited extent, which I'll show in a second. This would probably slow the reproduction rate pretty significantly. Um, but according to the ecologists in the working group, including Rich Lenski and Von Cooper, the advantage of reduced predation could plausibly outweigh the disadvantage of reduced food sources. Again, this is an uncertainty, uh, but it's plausible that this could lead to exponential population growth and allow mirror to expand and potentially invade diverse environments depending on the mirror created, or they could potentially evolve once they're released into the environment. It's unclear. Yeah. I see another question. What would be the infection mechanism of mirror if they can't interact with natural cells? How could they be pathogenic? This is described at length in the technical report. I'm not going to just send you over there because it's 300 pages, but um, the long story short is that there are a couple mechanisms, again, an uncertainty here, but they could consume nutrients to a sufficient level that it causes nutrient deprivation or some harm to natural host tissues. They might also cause some, eventually, there might build up to such an extent that they could cause some damage. And there's another pathway toward immune activation called damage-associated molecular pattern recognition. And so if there's enough damage signals caused by cellular stress, stress signals in cells also can trigger immunity. It's a little bit hard to wrap your head around, but basically if a mirror infection grows to such an extent, it might go under the radar for a while and then cause such damage signaling that you could have some sepsis-like. It's plausible that there could be some sepsis-like phenotype, which is basically a dangerous amount of inflammation once the infection has grown past a certain rate or state. That's my quick answer there. But it is a difficult question. It's a very unprecedented type of infection. Um, So one common question, you're welcome, is what mirror might eat in the environment. This is a good question because mirror would not be able to consume most natural chirality nutrients that bacteria depend on, but they could plausibly grow in both natural environments and in the bloodstream by consuming only achiral nutrients. Like the many examples are listed here in this table on the left. This is directly taken from the technical report. And data from minimal media experiments show that natural chirality E. coli is capable of growing exclusively with several of these molecules as an achiral carbon source in concentrations of roughly like 100, I'm not going to go through all of them, but hundreds of micrograms per liter, which is within the typical ranges of these nutrients that are found in natural bodies of water, soil, the gut, and the blood. Four of the more important potential niches to evaluate here, I think. E. coli can also metabolize a small number of nutrients of opposite chirality, actually a few amino acids and such. Depends on the strain. And mirror E. coli, depending on the strain or whatever mirror bacterium we're talking about, could be expected to do the same. Um, in addition, it doesn't seem feasible to reliably contain mirror bacteria in the lab once they are created. Scientists currently use biocontainment methods to create dangerous bacterial strains that could not survive outside of the lab if they escaped. For example, by engineering them to be dependent on nutrients that are only available in the lab, auxotrophies. Um, these methods could be applied to mirror bacteria. You could create a heavily auxotrophied mirror bacterium. But these methods could just as easily be undone using the same basic techniques by malicious actors over time. Um, and just physically containing mirror bacteria in high security labs unfortunately poses a non-trivial chance of accidental release from human error, which has an unfortunately well-documented history, or again, from deliberate evasion from malicious actors. And it's possible also that certain incentivized actors either misincentivize companies or malicious actors could use existing methods to make mirror bacteria more dangerous or environmentally robust to reduce costs or cause damage. For example, by giving them the ability to consume glucose in the environment. The pathway to consume mirror glucose is known. And so the inverted pathway could be easily engineered into mirror bacteria, which would give them a major food source and boost their reproduction rate as well as their chance of survival in the environment. Um, I see another question. Could mirror cells not just grow on achiral carbon sources, but also have racemeases to turn regular nutrients into mirror? Yeah, they could. It depends on which mirror bacterium has been created and does it have these pathways. It's probably unlikely that it would evolve those quickly, I think, but I'm not sure. But yeah, short answer is yes, depending. Finally, one more question that people often ask is whether countermeasures could mitigate this threat. Um, and I think it's currently unclear whether existing achiral or mirror image antibiotics would be effective against mirror bacteria. but they probably could be developed. There probably could be pretty good antibiotics that could be developed to target mirror bacteria and not natural chirality cells. Other novel countermeasures could potentially be developed, vaccines, etc. Um, However, countermeasures often actually require a functional immune response to be effective. People with severe immunodeficiencies struggle to survive infections even with extensive medical treatment, antibiotics, vaccines, etc. And it's not fully understood why this is the case, but mirror infection might be similar. It's not clear. Um, and it'd also be challenging, I think, to say the least, that developing, testing, and distributing a continual supply of novel antibiotics globally during a serious mirror bacteria outbreak, would be very difficult. And there is the potential that mirror bacteria could evolve resistance over time, just like natural bacteria or something else too. And finally, probably most seriously, is that medical countermeasures for humans can't plausibly prevent ecological harms, basically, to plants, animals, ecosystems. And so while these are good to consider and worth investigating for human use, ultimately, I don't think they will mitigate too much of the potential risk. So quickly summarized all of the risk assessment there. This analysis was published in two documents, a science policy paper in Science, and this accompanying 300-page technical report, which provided much more detailed analysis of the feasibility and risks, and the paper in Science kind of summarized these and also made a few preliminary recommendations to consider for next steps. To clarify my involvement, I worked to support the working group, but was an author on the immunology chapter of the technical report, not on the science paper. And I've since been working at MBDF, particularly engaging with and soliciting input from the SynBio community, like I'm doing in this very moment. Um, so one of the the key recommendation in the article was for a global discussion to evaluate and chart the path forward. And as a starting point for that discussion, the group put forward a couple tentative recommendations. One was that current applied work with mirror biomolecules is safe and should continue, as should ongoing work with natural chirality synthetic cells. Second, it recommended that given our current understanding, mirror bacteria should not be created, given the potentially unprecedented risks and limited potential benefits. And third, research should be conducted to better understand the risks of mirror bacteria, as long as that research doesn't facilitate the creation of mirror bacteria. And it should be done transparently to avoid security dilemmas, likewise for countermeasures. But there are varying degrees of buy-in on the seriousness of these risks, and future research can and should be done. Fourth, down here, society should carefully consider options for potential governance right now for the precursor technologies for mirror bacteria. Importantly, the article did not call for restrictions or moratoria on any ongoing research. I see another question. Is it possible that natural predators would learn how to predate on the mirror bacteria to naturally limit the infections? It is possible. I don't know how likely it is. My guess is that the selection pressures would favor mirror bacteria to, adapt to consume additional molecules more so than it would natural chirality bacteria, because mirror bacteria probably wouldn't be available to a larger extent, or if they're growing at a lower rate for a while. But I think they could over time. It's unclear how likely that is to happen, and it might be pretty difficult. But yeah, again, short answer is yes, possible. So nearly done with the background, well, not background, but the discussion since the paper was published. So there's been quite a lot of discussion since the paper was published, including this is a selection of venues that have discussed mirror bacteria in the last year. So I'll summarize just a couple of these, starting with a few examples of scientific discourse. So several e-letters were published in response to the paper, which you can read on the Science paper website, including one on the potential role of glycan molecules in immune recognition. This led to a really fun and productive roundtable at Glycobiology Conference Glyco 27 in Canada earlier this year, which I helped organize and attend. To briefly summarize several days of discussions with a bunch of glycobiologists and glycochemists, I think it's reasonable to say that the immune system might be able to recognize some mirror glycans and induce some level of inflammation, but that this would almost certainly be insufficient on its own for an adequate immune response, just because of how the immune system reacts to glycans, which is pretty minimally. And it doesn't really interact with the adaptive immune response too well. There's also a recent 2-day workshop on mirror biology hosted by NASM, um, the National Academies, which brought together a bunch of experts from government, academia, and industry uh, to discuss the potential risks of mirror bacteria. Um, what is this question? Um, Limited access, interesting. Is the mirror life human viable? I don't think so. Short answer to that one, I think is going to be no. Assuming you're talking about a mere human being, which I think is not likely to be made. Um, what are the other methods the community has to contain the creation of mere life besides scientific reports? I'll get there. Good question. Um, right, NASA workshop covered lots of stuff. A report will be published in the spring, but the talks themselves are available, I think now or very soon on their website. There are a couple other venues that have also taken up this issue and made a call to action. First, in January, the UK government's chief science advisor chaired a roundtable of experts, which recommended that mirror bacteria not be created and suggested that a broad effort was needed to prevent their development. At the Spirit of Asilomar Summit in February, an entreaty was signed by about 100 participants stating that mirror life should not be created unless the risk picture changes significantly. Um, Recently, a draft report from the UNESCO International Bioethics Committee, which includes Marilyn Dogterum, has proposed a precautionary global moratorium on creating mirror cells. And most recently, this is Germany's Central Commission for Biological Safety, uh, which has a working group on synthetic biology, also released a statement supporting the key considerations or conclusions of the science article and um, suggested that more discussion is needed. Um, Two other scientific discussions, which were co-hosted by MBDF. The Paris Conference on Risks from Mirror Life was co-organized by MBDF and Institut Pasteur and hosted at the Institut Pasteur in June. And here, over 300 stakeholders from more than 30 different countries came together to discuss the risks and feasibility of mirror life and potential need for governance. And based on these discussions, the conference steering committee shared the following recommendations. The first is that researchers should refrain from pursuing the creation of mirror organisms, and that public and private funders should make it clear that they won't support research aimed at this goal. Second is that work should start now to develop frameworks to govern key technical milestones on the pathways to mirror life. So I'm getting to Mariana's question here, or hinting at it, and then we'll talk about it more in a few slides. But third is that efforts to mitigate the risks of mirror organisms should preserve scientific freedom and potential benefits of the research as much as possible. And finally, further research should be conducted into the risks of mirror life as long as this research doesn't help create mirror life. But again, more research should be done. So stepping back a bit, it's clear from the discussion that there are still many unanswered questions and that new evidence can always change our understanding. So we should continue to try to evaluate the situation and learn more in the time that we have before the capabilities advance too far. So looking ahead, quite a lot of discussion has been happening and will continue to happen on point two, the development of frameworks for governing milestones on the pathways to mirror life that mitigate these risks. Again, goal, mitigate the risks, keep the benefits, protect the benefits of mirror biomolecule research as much as possible. And so this was the main focus of a workshop that just happened outside of the Engineering and Safeguarding Synthetic Life conference at the University of Manchester, which was held last month. This was a follow-up workshop to the Paris conference, which was co-organized with Patrick Tsai outside the University of Manchester last month, and focused on the synthetic biology components of the question and the pathway to mirror life. So the aim here was to ensure that any discussions on governance are grounded in technical reality, and to help make sure that policymakers in the future understand the benefits of key technologies. And so there were discussion of 4 key precursor technologies that were identified at the Paris meeting. So this was discussion of protein synthesis using recombinant elements or pure systems, mirror ribosomes, crossover translation systems, and synthetic cell natural chirality cells. And I'll discuss each of these in a few slides. But I think the key readout from the workshop is that everyone agreed that current work, like making mirror proteins and mirror nucleic acids, does not pose any direct threat or is a problem. They're not self-replicating, they're not a danger. The question being discussed is really where to draw the line between the current research and research that could enable or the creation of a mirror cell. And there's a lot of debate about that question, and that will eventually be shared in a report in the coming months. So prior to the workshop, we worked with the invited participants to develop a working paper to assess the current state of the art for the relevant technologies on the pathway to creating mirror bacteria. Um, the technologies discussed were identified at the Paris conference, and for each of the full list is here in this bullet list. It's the four precursor technologies that I discussed on the previous slide, as well as a few others, including mirror genomes. Um, and for each, the following characteristics were discussed. You had to define the technology, discuss the relevance to mirror life, the current state of the art, key bottlenecks, and then the beneficial uses, and then also alternative technologies that might achieve the same benefits. Um, which I think is important to consider. So the workshop participants collaborated on this document, which wasn't intended to be so long, but it ended up being nearly 50 pages, summarizing all of this. And this will be combined with the workshop proceedings into a final report. But I'd be happy to share it with anybody. So it'll be public eventually, but I'd be happy to share a draft version now with anyone who would like to read it. Let me know. My email will be on the last slide. Um, so the challenge of all of this is to identify milestones along the way that could be used as potential barriers or stopping points. So to simplify things greatly, you could imagine just a single linear pathway, which is not the case. But imagine so. On one end is research to create mirror molecules for therapeutic purposes. This is what we can do now. This is currently possible and should continue. On the other end is the creation of mirror bacteria in the coming years to decades, which I think should be prevented. Of course, once a milestone has been achieved, it becomes much more difficult to use as a stopping point. So right now is this rare window of opportunity to anticipate a future risk, discuss and identify valuable stopping points that the community can agree on in advance. Um, and again, I can't stress this enough. The ideal barriers would allow beneficial research to continue as much as possible, while also robustly preventing the construction of mirror cells, or at least making it so that anyone attempting to create mirror cells can be identified and prevented. Um, so outlining the pathways, because it's not just a simple linear pathway, uh, is pretty hard. It's a little bit difficult to, understand what the future might look like. And this has been a key focus of discussion recently and the main focus moving forward. So now I'd like to quickly walk through some potential pathways here and the current state of the art for a variety of technologies, which is heavily informed by this workshop that we just had. Um, so as I said, there are two most likely pathways to creating a mirror cell. There's the top-down and bottom-up pathway. I'll talk about the top-down pathway in a few minutes, but we'll start here by talking through the various precursor technologies that might be needed to build a mirror cell from the bottom up. Um, so at a high level, four things will likely be needed. First, a mirror DNA genome. Second, a transcription or translation system that's capable of generating a functional cytoplasm. Third, a membrane to enclose these two things. And finally, some method of combining them all and booting them up to create a living cell from non-living parts. So for a regular natural chirality cell, items 1, 2, and 3 can be easily accessed by taking apart existing cells. But what hasn't been accomplished yet is this booting method, number 4. Uh, Combining these things is not straightforward, and it had not been achieved yet. Yeah. So for the purposes of this talk, I'm going to mention 4 types of synthetic cells. I'll use the word synthetic cell, but this could mean one of 4 different things that could be created. And these most likely would have to be done in order really to achieve a mirror cell. So the easiest, not easy, but the simplest version, also simple, is a synthetic cell with cell lysate derived components. So it's important to note that this would include undefined components, just like full cell lysate. We don't exactly know every single thing that's in it or what concentrations or what modifications have been used. But using these components to boot up a synthetic cell would still be a really useful model for further experiments and would still advance understanding significantly and have demonstrated the potential for booting. Massive accomplishment. 2 would be a step higher in difficulty. This would be a cell from recombinantly produced components. So this would be defined on a molecular level. You have to know every protein and other molecule that you need to boot up a cell, but it could still have many undefined post-transcriptional or translational modifications of unknown structure or function. From there, and another increased step in difficulty would be natural chirality cells from chemically synthesized components, fully chemically synthesized components, which would have to be completely defined and understood in order to achieve this. Probably pretty massively difficult. And then a mirror cell could be made from the same mechanism as number 3, same protocol really, but using mirror image components, which could probably be synthesized just as easily, but probably more expensive um, due to availability, at least currently. Um, so again, the first cell type here has not been achieved, but many researchers are working on this, including people on this call. I think there's disagreement about how easy this booting step will be, but could be soon, could not be soon. Only time will tell. You all know this. I'm looking forward to the future. So, but to reiterate, going back to the mirror cell version, um, the mirror genome, the mirror cytoplasm, and the mirror membrane would all need to be synthesized entirely in vitro without the use of cells, none of which has been done before in mirror version. So assessing how difficult these steps would be is key to assessing how difficult it would be to create mirror life. So starting with a mirror genome and assessing the current state of the technology here, how difficult might it be to assemble a full mirror genome? Synthesizing short DNA oligos in mirror form is basically just as easy as synthesizing natural chirality oligos, but the reagents are currently very expensive because there's low demand. So researchers can synthesize full-length natural chirality genomes already by assembling oligonucleotides together. Um, but when this is done, cells are almost always used as intermediate hosts for larger DNA fragments because they're protected from damage and they can be amplified for later use in vitro. But assembly of large DNA fragments without cells is feasible, it just hasn't been done at the same scale. So on the right is a gel from Dan Gibson showing his method for assembling DNA in vitro, which only needs three enzymes, a polymerase, a ligase, and an exonuclease. Um, this could be easily replicated for a mirror DNA if you have mirror versions of those three enzymes. And he was able to use this to produce a 900 kilobase pair product, although again, cells were used as an intermediate step for amplification, which would not be able to be replicated in a mirror version. Um, to my knowledge, the largest DNA fragments that have been assembled completely in vitro without any in vivo steps were the 27 kilobase pair mpox genome fragments for this 2024 paper, which were then amplified in vivo before again being assembled in vitro into a full 200 kb genome. Um, yeah. Uh, and in mirror form, kilobase length mirror DNA and RNA has been synthesized using mirror polymerases by Ting Zhu's group. And as I said, a functional mirror ligase has been synthesized, but not yet an exonuclease. So in summary, I think methods aren't yet optimized for genome scale assembly completely in vitro, but overall, my understanding at least is that assembly of a mirror genome is plausible today, but significant efforts and money basically would be required to do it. I think this was the general assessment at the Paris conference as well, which is why the mirror genome wasn't selected as one of the four key um, milestones on the pathway to discuss. Mostly achievable. Um, so moving to protein synthesis. Um, as time has gone on, synthesis methods have been able to generate larger and more complex mirror proteins, which you can see on this chart. Y-axis is increasing protein size over time on the x-axis. I really wish that I had time to make these to scale because the larger proteins should be larger, obviously, but they're smaller on here, which kind of bugs me, but that's on me. I think it's fair to say that progress here is uh, mostly driven by improvements in natural chirality chemical synthesis, not mirror chemical synthesis. But as I mentioned, the natural chirality chemical synthesis methods can be directly applied to mirror peptide synthesis as well. There's just a much larger market and demand on the natural side of the mirror. Um, so current chemical synthesis can routinely generate peptides of like 60 to 100 amino acids, sometimes longer under certain conditions. But I think that improving this length in peptides that can be routinely created beyond this limit is difficult, although it is improving. But this increase in protein size over time, mirror protein size over time, I think has more so been enabled by the subsequent innovations here. So one key advance is the development of native chemical ligation by Phil Dawson, Steve Kent, and others, in which several peptides produced by chemical synthesis can be ligated together to form larger proteins. So this gets you like, a couple hundred extra amino acids, though again, larger versions have been achieved. And then adding an additional innovation, more recently, proteins have been engineered such that they can be split into different subunits, which can be independently synthesized and in, like, say, 300 amino acid chunks, and then co-folded together to assemble a larger protein without chemical linkage. So a recent advance here was the largest protein here, this T7 RNA polymerase by Ting Zhu's group, which is the largest mirror protein that's been synthesized to date, employing all three of these methods together. Um, so 900 amino acids is like quite large. This means that many proteins in the proteome are within reach of current capabilities of chemical synthesis, but This is still like extremely laborious and costly to produce large numbers of proteins, certainly the amount that you would need to boot a cell. Um, but to build a cell, as I mentioned, um, what ultimately is needed is a translation system capable of replicating itself. So in this case, a key part of this is a fully synthetic mirror ribosome that's able to make more mirror ribosomes. And right now, this is a pretty distant prospect, I think. Like cost, fidelity, and technical feasibility are all barriers because the ribosome means the synthesis of more than 100 proteins required for ribosome biogenesis in vitro, uh, among other plenty of unknown factors that might also be necessary. But I think progress is being made here on kind of two axes. There's in vitro ribosome biogenesis of natural chirality ribosomes and improving pure systems, both of which could contribute eventually to creating a mirror pure system. Um, I think for the sake of time, I kind of want to go through these somewhat quickly, but I'm highlighting here a couple of recent papers, recent advances. For example, in vitro biogenesis of a natural chirality ribosome was recently achieved by Wataru Aoki's group in Japan, and they did rely on an E. coli cell extract for the basis of the method, but the ribosomal proteins and RNAs themselves, or the proteins themselves were introduced, and I think some of the RNAs were native. But this is still a pretty significant advancement in vitro biogenesis, but still far from being able to chemically synthesize ribosomes. Um, and then again, as far as actual mirror ribosomal components, a mirror image T7 RNA polymerase was chemically synthesized, as I mentioned on the previous slide, and it was capable of transcribing the full-length mirror versions of the 5S, 16S, and 23S rRNAs in mirror form. Um, Importantly, these didn't have the modifications, like post-transcriptional modifications that might be important for full function, because those modifying enzymes haven't been made in mirror form. Also, a couple of the several ribosomal proteins have been made and co-assembled with the 5S rRNA. Um, so to summarize, ribosomes have only very recently been, the biogenesis of ribosomes in vitro has only recently been achieved. And the process is still heavily dependent on cell extracts. So several barriers remain before one would be expected to be able to synthesize one in mirror form. Um, I see a question. Is it possible to imagine some hybrid cell with some parts mirror and some parts natural stereochemistry? Would that hybrid cell also be dangerous? I do have a slide on this in a few slides, so I'm just going to push this question for a few minutes. Sorry. Um, so the other axis I mentioned is pure systems. Um, Pure, I think most people in this audience probably know this, but Pure is an in vitro transcription translation system that comprises a solution of ribosomes, polymerases, a variety of supporting enzymes and other translation factors to generate proteins from a DNA template. And they have a wide variety of applications, and generally are inefficient on the scale of they take more proteins in to make the pure system than they can actually output themselves. So a key goal, I think, for the field, one of the key goals is to produce a pure system that can achieve self-sustaining protein generation. So it can produce more proteins than it um, than it requires to make it. But this would also require the ability to produce more complex proteins like ribosomes, so that it can replenish the ribosomes in the pure system, which is quite difficult. And so that's why the ribosomal biogenesis is also important for this. Moving through this kind of quickly for time. But one key advancement here recently is Yoshihiro Shimizu, who is the original developer of the Pure system and one of the leaders in improving them. He was able to express a minimally functioning set of tRNAs with Pure, 21 different tRNAs, which is going to be a necessary subcomponent of any self-replicating Pure system, and it will have to replenish the tRNAs, but still would require many other things. Interestingly, I also thought Ashti Karim, if I'm pronouncing his name correctly, his Build Us All talk, like I think it was 2 weeks ago, was great and showed the importance of improving metabolic function for increasing translation efficiency. So lots of people are working to improve the efficiency of pure systems here. And yeah, so I'll only briefly touch on lipid membranes because they're not likely to be a barrier. Basically, all the mirror image chiral lipids for existing bacteria can be chemically synthesized quite cheaply, and liposome technologies are advanced enough. So it's not expected at the Paris conference. I think people generally agreed that this is not expected to be a significant barrier to creating mirror life. Um, and so cell booting is like this weird mystical fourth step. And I think it's probably one of the more difficult steps to predict what is needed to bring non-living components to life. We don't know yet until we really do it. Some think it'll be really straightforward, some think it'll be extremely difficult. But I think recent progress indicates that it should be pretty achievable. Again, going a little bit quickly, some key advancements are John Glass and JCVI's work on genome replacement and transplantation. Kei Fujiwara's group was able to combine a minimal E. coli genome, a lysate-based transcription translation system, and a liposome and they were able to achieve some protein expression from this. Of course, nothing like being able to generate a full cell, but still, that's the basic formula for cell booting. And then a 40 kilobase per T7 bacteriophage genome was also successfully booted in a test tube, which is cool. So um, moving on. So back to the question of like what it takes to build a full mirror cell from the bottom up. I think synthesizing a genome is probably technically feasible today, but there's some serious barriers, and it'd be extremely expensive to accomplish. You'd need to synthesize a mirror exonuclease, and it'd still need to be assembled without in vivo amplification or sequencing, which is probably surmountable, but not straightforward. Uh, then a mirror image transcription translation system, as I described somewhat quickly, is probably quite a bit more difficult to achieve, and probably the most significant remaining technical barrier. Mirror membrane is probably not a significant barrier. And then the booting method is this like unpredictable, unclear um, uh, barrier or milestone that we don't really know the answer to yet. So yeah, put together, I don't think there's like a fundamental barrier to the bottom-up creation of a mirror bacterial cell. Um, and yeah, we'll have to keep monitoring progress here. So here's the stepwise conversion of top-down pathway. So Kirsten, your question here, I hope is answered to some extent. Um, I'll walk through this quickly. Um, so again, this is the idea here is to create, starting with a natural chirality bacterium, adding mirror biomolecules and mirror transcription translation systems, such that you can create this over time a mirror image protein synthesis capabilities within a natural chirality cell, and then eventually turning off a natural chirality component of the cell and converting the entire thing into a mirror bacteria. Um, this would probably be really complicated and would face a lot of the same barriers as as a bottom-up pathway, but also have some unique barriers in addition. Um, Primarily, it'd require engineering normal natural chirality ribosomes to efficiently make and extend or make mirror proteins, translate mirror proteins. Currently, they're only able to incorporate a few amino acids at a time, and I mean like 2 to 4 amino acids. Maybe 7, I think, has been achieved, let alone hundreds that would be required for full mirror protein translation. So that's a difficulty. Then you'd also have to enable natural and mirror protein synthesis within a single cell, this hybrid cell that Kirsten is asking about. Um, this would require genetic code expansion. You'd have to free up codons for the mirror amino acids, as well as you'd have to mitigate off-target interactions of these two orthogonal, well, hopefully they're orthogonal, transcription translation systems within the same cell. Um, and this was discussed quite at length at the Manchester conference, but so there's some thought that the mirrors system could be sequestered in an organelle or some other part of the cell, as Kirsten suggests here. Um, but it's still pretty hard to imagine having these two functioning very well in the same cell and then transitioning from one to the other while mitigating toxicity for the cell at every stage along the way. So for those reasons and others, most of the experts that we talked to thought that this was like more difficult to achieve than a bottom-up pathway, but that's not certain and it's hard to predict the future, as I said. So it's worth keeping in mind. Um, as for Kirsten's question, would a top-down cell, would this hybrid cell be dangerous? This was discussed quite a bit at the Paris conference because it's possible that a crossover cell that can do both could mitigate the need for a mirror bacterial cell. It could produce mirror biomolecules for therapeutic uses um, without having to create a mirror cell, which could be great. But others do think that it would be similarly risky. Some think it'd be more dangerous because it could still have access to natural chirality nutrients. But then there's the question of would it have the same immune and predation evasion? And I think the shortest answer is I don't know. My sense is that a mere bacterial cell would be more dangerous than a hybrid cell, but that's far from certain, and that's mostly my opinion, which I think is somewhat informed, but not maybe completely. So one thing, just very quickly on this slide, Some people often point out that the first mirror cell is going to be really feeble and not dangerous, and I completely agree with this. But I think the important thing is that if we get really good at booting cells, someone somewhere might boot a more complex and more dangerous cell. And so once we have one mirror cell, we might have many mirror cells, and some might become very, very dangerous at some point, and that's a key concern. So quickly, next steps are like building on this working paper from the Manchester workshop. And that basically involves soliciting input from a variety of different communities to evaluate the difficulty of achieving each step on this pathway, each milestone. Basically, by soliciting input from the field on estimated technical difficulty, potential costs, timelines, and incentives for each milestone, to eventually evaluate where lines might be drawn on such a pathway to maximally protect benefits, minimize risks. And then I also mentioned quite a few times that the available evidence about the risks is pretty limited. So what we're doing in parallel is trying to work to deepen our understanding of the risks and figuring out what evidence could change this assessment or create a better understanding of the degree of risk. So wrapping up quickly here, our funders and a variety of science philanthropies and our MBDF's advisory committee, all of whom are a real pleasure to work with. And this was pretty quick. Yeah, with that, I'd be happy to take any questions. I know we're like just at the end here. I really am keen to learn from you all, so please have a low bar for emailing me with any future questions or if you want to read the working paper. I put some of the questions up that I think are important and good to talk about, but happy to take any others. Thank you all for your attention.
Thank
Sorry,
you
it was a
so
little fast.
much. Thank you so much. This was really informative. I think a lot of people are having a hard time wrapping their heads around this whole project because it's such an unusual framing of here's this cool research and we want to do it and why.
Yeah.
Opening the floor for questions if anyone has any outstanding questions that haven't been asked yet. I just want to say, oh, I think there's a question in chat, but I want.
Trisha asked, so nobody is working on a mirror cell right now, and
Oh,
to my knowledge, that is true. No one is claiming to work on a mirror cell. I think if someone did claim that, they would be under some serious pressure. And I don't think anyone is working on creating mirror bacteria right now. And certainly hasn't announced it. Jan asks, has mirror life happened before on Earth? How would we look for clues? This is something that's been brought up quite a bit. I think it's extremely unlikely that life arose twice somehow, and some was in one form and of one-handedness, and one was the other, and both continue to persist. I think it'd be pretty easy to look for because you could do mirror PCR or something to try to identify or like amplify mirror nucleic acids in environmental samples. But the likelihood, if it does exist somewhere on Earth, is that it's like extremely rare and maybe in only some deep sea vents somewhere or something. I don't know. So we probably wouldn't be able to find it very easily. So I think most likely it does not exist somewhere on Earth. Maybe somewhere else in the universe. I don't know. Jose asks, sorry if I missed it, but why did we want to build mirror life in the first place? Are there any benefits we're giving up, or was it just research? I did have a slide on this, but it's okay. I went through many slides and no judgment. The main use to summarize is that mirror biomolecules do have some therapeutic benefits. Mostly they have a longer half-life than natural chirality molecules, and they are also non-immunogenic, which again contributes to the risks of mirror bacteria, but is not a risk in just molecule form because they can't self-replicate. So mirror bacteria could be used. The proposal is that they could be used to biomanufacture mirror biomolecules at scale pretty cheaply. But there are alternative ways of producing them. So that's the main proposed mechanism or reason for producing mirror life, I think. Good question, Jose. It's really the key question. Yeah. Any other questions? These are great. Keep them coming.
okay. I think we're getting to time, and I see some
We are
people
at
are
time.
logging off already. So I just want to say thank you so much, Ryan, for giving the
Thank you,
amazing
Kate.
overview. I realized there was a lot of information, and I think it's super useful for people who are new to this topic.
It might have been too much information. But thanks, everybody. I'm happy to hang on, or again, email me. And yeah, thanks for coming.
Thanks, everyone, and thank you for great discussion too. And have a great rest of your week.
Thanks, Kate.
Bye.
Bye.